My project is on the evaluation of patient-derived xenograft model accuracy using differential gene expression analysis for optimization of cancer modeling. Patient-derived xenografts are a form of in vivo cancer modeling for personalized medicine techniques. So personalized medicine is a novel method of treating cancers that has been developed through extraction of patient tumor tissue and then testing of specific treatments using tissue models. Traditionally, 2D in vitro cell culture models have been used for this treatment testing. However, those don't always accurately reflect the workings of organismal functions and therefore don't always present the most accurate results of the treatment. Instead, these new PDX models are samples of tumor tissue that are transplanted into immunodeficient mice hosts where the tumor is allowed to grow, and the treatments that are used on these mice more accurately represent the varied organismal function through the multitude of genes that are present in animal organisms. So I am focusing on these PDX models regarding colorectal cancer. Colorectal cancer has 4 specific consensus molecular subtypes, each that express different biological traits. And I am analyzing which genomic trends are affected by subtype differences in these PDX models so that we can analyze the effectiveness and accuracy of the models for future use in personalized medicine. I did this through public access data from the PDMR database from the NCI and various R packages listed on my poster. So my procedure involved extracting gene isoform files from the database, then performing differential gene analysis procedures using the DE-seq2 package. And then after that, I classified all of the different consensus molecular subtypes for each of the patients in the database, which after I extracted my counts file for my differential gene analysis, I performed various analyses to express the results of this data, including an Euler plot, volcano plots, gene set enrichment analysis, and also principal component analyses. So first, I, using the differential gene analysis data, constructed a bubble plot displaying results of the gene set enrichment analysis. This is done by organizing the human genome into hallmark gene sets, which have different functions such as development, immune function, and metabolic function. And this shows the FDR is the significance. So all of the points on this table are significant because their FDR value is lower than 0.05. And the NES, or normalized enrichment score, shows the difference in expression between the PDX and the tumor models. So you can see that the CMS1 has positive NES values, meaning upregulation of specific metabolic functions compared to the for the PDX versus the tumor. And also, another thing you can see is the epithelial mesenchymal transition in the development functions are downregulated in the PDX compared to the tumor, which has consequences that I will describe later. These principal component analyses serve as a visual representation of the DGE data, and these show that the tumor and the PDX data have they're grouped in different clusters, which shows that they're very distinct from each other. Next is these volcano plots. So these are used to identify statistically significant genes with high fold change magnitudes, which means the difference between the PDX expression and the tumor expression. So genes that lay on the two extreme edges, so the left and the right edges, are the genes with the greatest magnitude of change, while genes towards the top of the graph are the most significant. So the plot on the left represents the overall PDX versus tumor expression regardless of subtype. So we can see that most of the fold change is negative, while the CMS for PDX versus tumor plot shown on the right shows that there are both extreme positive and negative values, and also a few points that are extremely significant. And lastly, the Euler plot is used to quantify differentially expressed genes in each CMS. So it'd be one comparison of tumor versus PDX and another of across the subtype. And you can see that this subtype 4 has the most differentially expressed genes. So using these results, we can see that, as I said before, epithelial mesenchymal transition downregulation in the PDX model could cause potential treatment errors. Also, apoptotic functions have discrepancies in regulation that also would affect the treatments, which is not potentially dangerous because the treatment results in the model are not equivalent to the ones that could be used for the patient. And also, the volcano plot shows a few specific genes such as CLL4A2 that are significantly differentially expressed, which could be linked with regulation discrepancies and should be investigated further. So in conclusion, gene regulation discrepancies revealed by this study should be subject to further investigation of factors because these differentially regulated genes could have large impact on the success of PDX modeling. Because of previous modeling errors in traditional in vitro cell culture models, this optimization of specific differentially regulated genes in patient-derived xenografts should have extensive benefits for those patients undergoing colorectal cancer treatment. Thank you.